Yes, so I'm going to talk to you about a project we've been working on over the last 2.5 years, and it's really a case of trying to understand how the environment interacts with disease occurrence. So we've been working with available data and trying to apply species distribution modeling techniques to see if we can reveal anything about these potential relationships and correlations with environmental variables. We're then going on to look at making predictions in terms of where disease may be occurring across the country, where prevalence may be different. This is always much easier if you have direct surveys, and part of what we've been doing in this project has been working with other types of data. So things like Tree Alert, which is hosted by Forest Research and gathers data from landowners and members of the public, and new disease outbreaks. So these are really useful things for policy and for designing surveys and for framing questions and hypotheses for future experiments. So hopefully I also have control. So firstly about the aims and scope of our project, and really it is, as I said, applying the different modelling techniques to try and predict current and potentially future distributions of pests and diseases. We applied this to 3 case studies, each of which different biological organism, but also a different data set available. So ash dieback, we only have Tree Alert data, so this is self-reported data. It occurs as and when people choose to send it in. And we wanted to see if we could work with this data, which we assume has biases within it. Dothostroma, where we have lots of Forestry Commission surveys, the districts went out and looked for signs of disease. And Phytophthora pluvialis, which has arrived recently to Great Britain. So we have the initial surveillance that's been conducted, but this is not a design survey. Really, they're going to try and find occurrences of disease where they happen, and so are really targeting in on specific areas. And so each of these has a different challenge for modelling and working with the data. So on to an overview of the talk. Firstly, just going to set the scene and talk about the disease triangle, talk about how the environment can interact and affect disease distributions. I'll give some background, which is our motivating example, acute oak decline. This was a previous project we had worked on that has quite strong predisposition links to the environment. So drought in particular is important there. And then I'll highlight 2 of the case studies where we've used similar modelling techniques, looking at point process models. So this is looking at the density of reports and trying to associate those with various landscape features. So hopefully there's a theme there in what we're presenting to you. So the disease triangle is really quite a simple concept. It's just trying to widen out our thinking of disease. It's not just a pathogen happening on a tree, but for disease to happen, you also need a susceptible host and a conducive environment. So there are these 3 elements that need to come together for a disease to be expressed. And really, it's this environment area that we're focusing in on. But obviously, all these things are interlinked. So for the AOD example and predisposition, the environment can influence whether a host is susceptible. And we'll discuss these concepts with each of the case studies, because it's really taking correlations from statistical models and trying to check that they have some biological sense. So our motivating example is acute oak decline. It's something we've been working on for quite a long time, and we've published some prior work to the CFP project looking at this. So to give a background, this was something that first started being reported and named in around 2009, and it has 2 really clear symptoms that landowners were spotting, and not reporting into tree alert because that had not been set up yet, but the precursor to tree alert, the advisory service at FR was receiving an increasing number of reports of these symptoms, and it was unclear what the cause was. So just to discuss the symptoms, these are some clear images. It's this black liquid running out from cracks between the bark plates, and these can either be individual points or quite numerous on the stem. And this is often referred to as stem bleeding. And these emergence holes of the Agrillus beetle, which, as you can see next to the pencil, can be quite small and hard to spot. So for anyone wanting to go out and look, there are some key things to distinguish AOD from other similar diseases. Firstly, with the emergence holes, they're pretty small, but they're really distinctively shaped. So one edge is perfectly flat. It can be in any orientation, but we'll always have this really sharp, clear edge like a capital D. For the stem bleeds, like I say, these can be individual or in quite large number. Very few other things you could confuse with, but there are a few other, mostly root diseases, so Armillarium phytophthora, that could also cause stem bleeding, but the patternation on the stem is very different. So Armillaria, we have to think of it as a single organism growing up from the roots, and it affects a single area of the trunk in quite severe scale. So whereas AOD, we might have individual stem bleeds occurring in a random pattern, here we have a specific area, and almost every crack between the bark plates will have some sign of black liquid. So, as I mentioned, this was first described in 2009 with a sort of unknown cause. So colleagues at Forest Research, notably Sandra Denman, led research to try and identify causal organisms. These symptoms were seen as a unique set within the wider oak decline complex, but they match descriptions from across Europe dating back throughout the 20th century. But they're quite often associated with Agrillus infestation. However, when pathologists from Forest Research went and looked, they didn't find the things they were expecting. We might find phytophthora, but found lots of bacteria. So lots of work went on. Notably, Carrie Brady and then James McDonald have been working on these species to identify and look at causes. So it seems it's a bacterial pathobiome, so it's multiple bacterial species that interact together, and they're within that Brunneria goodwinii is probably the key species. But having the different species together increases lesion size. Having the native beetle larvae, the Agrillus bigotatus, present also results in larger lesion sizes and causes these increases in bacterial activity. So James has been working with Maureen Cambon, who's done some really nice work there, looking at how the growth of the bacteria increases in response to frass from the galleries of Agrillus bigotatus. And so this is quite a new set of research, but linked into a longer series of work on oak decline. And as a decline disease, predisposition from the environment and from other outside factors is an important driving factor in where disease occurs. So you can see the distribution here. It's southern in England, but from the East Coast up to the Welsh border. And recently, in the last few years, we're starting to get a few reports just over the border. So perhaps some indication it's expanding, but quite a clear, distinct area where we think it's probably filled out its environmental niche. So as a decline disease, scientists have come up with a model, and this is the decline spiral first developed by Magnon. And really it has three stages. So predisposition, as I mentioned, is the first stage. There's some initial weakening of the tree needs to occur. And this will often involve the environment. Drought is quite often cited as a cause. Soil conditions, other local environment issues can also cause the trees to be growing in suboptimal conditions. Then a second stage where inciting factors, these are more one-off events, so particularly severe drought year, particularly hot temperatures, as storm damage can cause zooming through my slides. There we go. Can cause a tipping point before finally these contributing factors, the bacteria and the beetles, can move in on susceptible trees. So predisposition here is thought to have a crucial role. So this was our sort of driving force between looking at the distribution and seeing if there are links to the environment. So we were aided in doing this by funding from DEFRA over 2 years to conduct surveys. So in the first year, in 2013, we looked across England and Wales quite widely in a completely random survey. And then in 2014, we focused in on the known distribution, which at the time is shown by these black 10-kilometre squares, and tried to assess how well the self-reported data had described the distribution. So we put a buffer around all these points of about 75 kilometres, and this is the largest distance between any 2 points in the distribution. We then selected all the 10-kilometre squares that intersect with that buffer. We got oak distribution information from the National Forest Inventory, so we knew where there were likely to be more oak woodlands, and divided it into a 50-kilometre grid where we adjusted the number of samples to the amount of land within the buffer. So we were selecting relative to land cover, relative to oak cover, and in a focused area around the known distribution. So we conducted our survey sample, got a random selection of 10-kilometre squares to go and look, and then worked with lots of local groups. So firstly, we used NFI to target in 2 sites, but we could not always get landowner permission to survey. So we worked with National Trust, Woodland Trust, Wildlife Trusts, Natural England, and lots of councils to identify oak woodlands we could go and visit. So that left us with a designed survey that we could split in lots of different ways to conduct our analyses. We could either have a full data set, very biased to where occurrences were happening because we had all the tree alert reports included. We could have survey-only data sets, or we could focus in on just this 2014 survey. And we can conduct our modelling through the different data sets and ended up with a very similar set of environmental variables that came out as being important. So during the project we're working on for CFP, we've expanded this to include oak condition survey sites across Scotland, as well as some extra sites where Forest Research have been working and observed no signs of disease. So we can reconduct the analysis with a wider data set and find that the same trends are holding, but get less error around these trends. So we can find that temperature is very important. So we're using a day degree model, which is quite often used with insects, and 11.5 as a cutoff threshold, as that's relative to the Agrillus beetle. So the warmer areas are, the more likely AOD is to occur. We can also put in rainfall, and as we're expecting, lower rainfall areas, more likely to be drought affected, are coming out as areas where AOD is more likely to occur. Similarly, low elevations and higher nitrogen deposition areas are also coming in as important variables. So we can use this to make a prediction across Great Britain, which is shown on the right-hand side, showing how likely we think AOD is to occur in different areas. So this is useful, but beyond that, we've used this to shape hypotheses for further experiments. So we've had some drought manipulation experiments to look at how lack of water influences AOD occurrence. And we have conducted lots of within-site sampling, where we're finding nitrogen is also having quite a key role in driving where AOD is and where it isn't happening within a site. So that was our motivating example. But for CFP, we wanted to extend that to new data sets. So the first one is ash dieback. And again, I'll just follow the same format and give a brief introduction to the symptoms and signs and history of ash dieback before going in to talk about the data we're working with. Notably, we don't have a designed survey to complement this analysis. So that was our big challenge. So ash dieback, Hymenecyphus fraxinea, it's a fungal pathogen that was first discovered in 2012 in Great Britain, but had been found in Europe over the decades preceding that. Two really clear symptoms associated with ash dieback, and the first thing you will probably notice is crown dieback. So it's a pathogen that infects the foliage and causes dieback from the tips. But this is a symptom that can be caused by many things. So for confirmation, you'd want to look more closely and see often these diamond-shaped lesions that form around the twigs. And so these would be key signs to look for. Also, you may see evidence of infection on the leaves, either small black marks, or as it progresses, the whole leaf and petiole may die back. And if you're looking at the right time of year, very carefully, because these are very small, you might see the fruiting bodies on the petioles. But yeah, these really are small to see. So in terms of a history, ash dieback was first reported as an issue in Europe in about the 1990s. And then quite a long period of research and spread throughout Europe has led to quite a bit of knowledge now about what may have happened. So this is thought to be of Asian origin and introduced possibly as early as 1978. There have been herbarium specimens where ash dieback has been found. So this is long before detection occurred. In Great Britain, as I said, first found in 2012, but it was likely present here from around 2004 and possibly longer from looking at the dendrochronology of trees that had died. Most recent estimate seems to suggest around 50% of trees may die over 30 years, but there is some level of resistance, and maybe we'll hear a little bit more about that later. But there's also indication that the environment can play a role, particularly in sporulation and infection. So we had good reason to start looking and seeing if there are associations with environmental trends. So, as I mentioned, our data is solely from Tree Alerts, which is the website Forest Research runs, where landowners, members of the public, anyone who's interested can report diseases freely and receive a diagnosis from professionals. So we're working with a data set that's reported at random, but all records have been verified. There's no survey. There probably is some bias. The first thing we can see looking at the map on the right is pretty much everywhere, lots of points across the country. But if we move that to a density plot, just very simply, we can see more are occurring in the south and west. What we don't know is if we can trust this, because there is likely reporter bias within the data. So, for example, Observatory is a program Forest Research runs. They train about 200 people to go out and look for disease. Ash dieback is something they report, but they'll only report it in if it's not already present in a 10-kilometre square. So we're already thinking we might have some levels of bias within the data. So our first step was trying to handle as much of this bias as we could. So we're looking at the total reporting through Tree Alert now and counting the number of different species that are reported in each 10-kilometre square. And we can see roughly there's a similar bias south and west. More reporting occurs. It's definitely got some links to urban areas. So West London is particularly high in the number of species that gets reported. And if you can look very closely in Kent, there's one square which is particularly dark red, and this is Bedgebury, where I think there's lots of effort gone into finding out every kind of fungus that's occurring there. So we know there's bias. What we want to find out is whether this has a link to ash dieback. So we can look at which of these squares have ash dieback reported within them, and we can see there's a really, really clear link. The more species that get reported, the more likely an ash dieback report is to occur. A second thing that we think may be important is the ash distribution, which again, if we look at the map, you can see darker green, south and west. So following a similar pattern, perhaps. So this is a map, it's an estimate, a guesstimate from a Smarties project. So this is something Rothamsted Research were involved in, looking at various factors affecting ash, including ash dieback and emerald ash borer. As part of that, we made a 1 kilometre estimate of the amount of ash. So we used broadleaf cover from Copernicus and then divided at the 20-metre scale the country into 5 land use classes, either Forestry Commission, woodlands, which are larger than 5 hectares, small woods, urban, and then just a blanket open countryside. And for each of these, we had different estimates that varied depending on region or country for the amount of ash within broadleaf woodland. So we could use things like the Forestry Commission subcompartment database, iTree surveys, countryside survey, and the National Forest Inventory. And so it's a guess. At the very local scale, it's going to be wrong, but hopefully at the broad scale, this will reflect the amount of ash that's likely to occur. So these are covariates we can include in our modelling, along with various environmental factors. So we're using maximum summer temperature, annual rainfall, and this geomorphons approach to dividing terrain. So this is taking elevation data and looking to assign valleys, slopes, peaks, and flatter areas. So it divides into about 10 categories, but we've grouped those together. And we can see our prediction follows roughly a similar pattern to the overall data reporting. It's denser in the south and west, and we've got this one outlier point where Bedgebury is. But the thing we can do next is remove those things we think are reporter bias from the model and just predict the environmental effect. And here, unsurprisingly, we are finding ash dieback in most areas of the country, but there still is a bias south and west. So the highest areas, which are sort of bright whites with a few individual red squares, are in the south and west of the country, and we're finding a strong link with valleys. So these are areas where moisture might be higher or remain higher throughout the day and aid infection. We should also bear in mind these are probably areas it might be easier to report from, so there might be some bias remaining. But I think that although there's potential for bias, it makes biological sense that moister environments might result in higher infection from ash dieback. We can then look at how this model might predict ash dieback occurrence in different climate scenarios. So we've got quite a lot of things shown here. Really, there's 2 scenarios. One, which is 4.5, it's fairly moderate, but it does involve us cutting emissions quite a lot. And then 8.5, which is really business as usual if we keep emissions similar level to they are now. So CEH has produced lots of maps and predictions for these, so we can get various environmental variables that we expect might happen at different time slices. So on the left, we have 2000 to 2020, then we have around 2050, and then from 2060 to 2080. So occurring now, slightly into the future, and in about 50 years' time. We can see the pattern changes. So if we're in the west, if we're in the north, the warming and wetter climates may increase the amount of ash dieback that occurs. If we're in the south and east, particularly in these high emissions scenarios, perhaps it may be less conducive to ash dieback occurrence. At this point, it's really worth pointing out the caveats to this. This is useful for informing future surveys, future research, trying to make a best guess for future policy. We're working with point process models. These are not looking at time as a specific factor. So it's really taking our estimates of the current environment and trying to look forward. And so, yeah, there's room for error here, particularly as temperatures in the south and east are not included within our sample data. So we're getting warmer than we would expect. We can do some sense checking. So Forest Research has recently made an online tool for climate matching. So it's aimed at guiding which species and provenances we might want to plant. But we can see the area of East Anglia we're talking about, where the little tree occurs. Its climate in 2060 to 2080 is likely to be similar to northern or southwestern France. And we know southwestern France, we're not getting very much ash dieback reported. So it's a believable prediction, but we need to keep our wits about as we're considering this is true. So I'll now move on to the second case study I want to talk about, Phytophthora pluvialis. Again, this is mostly a foliar disease, but it will cause lesions on twigs and stems, as you can see in the images. This is something first reported in 2013 in Oregon, affecting tan oak and Douglas fir. Is sort of considered probably in its native range there. Also affecting Pinus radiata and Douglas fir in New Zealand, this red needle cast. So quite a problem there. First discovered in Great Britain in 2021, but quickly found in Cumbria, Devon, Gloucestershire, Herefordshire, quite widely along the west coast of Great Britain. Affecting both Western hemlock and Douglas fir. The key thing to look out for if we're wanting to spot this is needle browning in the lower crown. So again, some evidence that climate impacts this. It's something that varies in its severity between years in New Zealand. Infection linked to moister, more warm rather than hot times in the autumn and spring. Like I say, it's mostly a foliar disease, but these needles are then lost. So regrowth can occur without infection. So something is triggering the severity of disease between years. In terms of the data, this is a quarantine organism and ongoing outbreak, so we can't really report or show you the data we're working with, but these are the plant health demarcated areas for Phytophthora pluvialis, and it's very westerly in its distribution. So we can then conduct similar modelling to the ash dieback modelling, and here we find summer temperature, very important, role of elevation, annual rainfall, also important, but here minimum winter temperatures that come out and really are the factor that help balance out the model. So not something we're necessarily expecting to find, but I think makes sense in terms of spore survival to between years. So our model can be compared against quick models that were done for the pest risk assessment, looking at rainfall and temperature, and broadly similar areas, but obviously quite a bit more detail within the predictions we've developed. I guess the question remains, there's things we need to test to confirm that these correlations are the true driving factors, particularly for this data, because it's very focused on outbreak sites. So we're making an assumption here in our analysis that plant health have looked everywhere across Great Britain, and what we know is the actual distribution. I think there's lots of helicopter surveys, lots of effort that goes into these plant health surveys. So I think we can have a reasonable assumption there that there's quite wide effort going into looking for this disease and a host species that's not very widely planted, so we can target in quite specifically for these surveys. We can also do similar predictions looking forwards. And again, we can see high temperatures have this similar negative effect in the south and east. But generally, warming, and especially warming winter minimums, can really increase the spread further north. So this is sort of in contradiction to the acute oat decline case study that we started with, where drought was a driving factor. And there, the south and east was the area where we would expect to see most problems. So different diseases influenced by the environment in different ways, and this is perhaps useful to inform future research and policy. So in summary, really the key finding from all of this is that the systematic survey really helps to have more confidence in these correlations. But there's lots of ways we can work with large data sets like tree alert to understand the bias, particularly if we have detailed host maps. And where there's lots of plant health surveillance, we can make assumptions, and potentially the models you'll make very quickly after something arrives are not going to be the most accurate, but they could be very useful for guiding further surveillance. Really to have more confidence in the long-term predictions, observations from a wider area, so Europe-wide, global, would really help. And yes, I think that's all of the points I wanted to summarize. Yeah, just this new arrivals really does come with lots of uncertainty. But yeah, if you're wanting to know other places in the country, it might be useful to look. It can be really useful for surveillance design. That was my final slide. I don't know if there's still time for questions.
Thank you, Nathan. That's really fascinating. Yes, we've got time for a couple of questions, and there are a few in the chat. What would be great if we don't get through them all is if you could perhaps answer some of them in the chat as well, because they're coming in.
Yeah, I'm just seeing one that's popped up, which was something that I had meant to mention, that our ash dieback predictions may suggest that the area is not so conducive for ash dieback, but that doesn't mean that it's going to be good for ash growth. So hot and dry will have other impacts on the host beyond the disease occurrence. Yeah.
So we have a question from Rachel Riley, which says that with the increased occurrence of ash dieback in valleys, is that valley bottoms or slopes? And if it's slopes, does it vary with aspect?
Aspect didn't come out in our analysis as important, but potentially it would if there's going to be a degree of error within the data we're working with. So the geomorphons breaks the environment or the elevations into 10 different categories, and we grouped the ones that were valley bottoms. They were the most likely, but we also had 3 other categories within the model that were fitted. So slopes and tops and flat areas. And it was really the flat areas were least likely, biggest difference there. Valley bottoms, highest occurrence, and slopes were somewhere in between.
Thanks. A related question is predicting less ash dieback in a warmer southeast England based on the climate of the middle of France. But what is the abundance of ash in southern France?
Yes.
Oh, sorry, this is the one you answered, isn't it?
Answered. So yeah, that was something I meant to cover.
Yeah. Thomas Sim says, do you have similar models for Dothystroma?
Yes, we have similar models for Dothystroma. There, we were slightly unsure if we were modeling coarse compine distribution or Dothystroma distribution, because it's occurring most places where coarse compine is. And we have some Scots pine models. Again, we're just starting to refine.